Hi, my name is Steve Lakiten. I'm a postdoc at the Blue Brain project at EPFL. My research focuses on sensory information processing for decision making. I'm very happy to be here presenting for the third time. And today my talk will be about cortical cell types, layers, and how they differently shape the geometry of neural representation in a biophysical model of the neocortical microcircuit. So a central problem in neuroscience is understanding how the neocortical microcircuit processes stimuli for perceptual decision. Generally, theoretical models of cortical network are mathematically convenient tools that have produced deep insight into computational principles. Yet, we argue that their lack of realism does not allow them to fully probe the role of the cortex biological diversity into shaping sensory representation for perceptual decisions. So here we address this question using this biophysically detailed model of the rat's primary somatosensory cortex. The model comprises about 30,000 morphologically detailed neurons, spanning all 6 cortical layers and receiving inputs from thalamic fibers. And the model also captures the biological diversity with 60 morphological types grouped into inhibitory types on the left and excitatory types on the right, displayed here. It features a realistic synaptic connectivity, short-term plasticity. It also captures 11 electrical types. And what we did here in this study is we simulated the response of this microcircuit to whisker deflection in 360 different orientations. And then we analyzed the geometry of these representations using a very recently developed and very powerful dimensional reduction technique developed at EPFL by the Mackenzie Mattis lab. So here, focus on the left of the panels. We first look at the sensory representation per cell types, and to do that, we train Zebra, that model, separately per layers, and using the cell synaptic types as auxiliary labels to shape the embeddings. We first found that the embeddings were clearly clustered if you look at the red dots representing excitatory cells responses and the green dots representing inhibitory cells responses, and this was consistent across the layers. These indicated a very different response pattern for this different population. We then looked at a different question. We trained Zebra this time jointly across the layers to find the shared latent space on which we could compare the embeddings. And we first found that for excitatory cells, the embeddings from the input layer 2, 3, and 4 overlapped while they became clustered, and these are the colors for layer 5 and 6 in blue and green, when we looked at the output layers. And this clustering was not observed at all for inhibitory cell. This is a lower panel on the second columns. Then we probed the sensory representation, that is the stimulus representation, in the different cortical layers. And to do that, we trained Zebra this time separately per layers and per cell types. And we used as auxiliary labels the stimulus direction and the trial IDs to shape the embeddings. And what we saw is that the embedding smoothly reflected stimulus directions. And when we looked at how well we could decode the stimulus orientation from this embedding, we saw very good decoding for the output layer 2, 3, output layer 4, 5, and 6 compared to the input layer where it was worse. And this result also holds for inhibitory cell, but they show poor decoding accuracy on the bottom panel than the excitatory cell. Overall, if we pull all the cells together, we get even better decoding up to using this cross-validated KNN machine learning algorithm. The decoding error goes down to 0.16 degrees. So in conclusion, the model we have predicts that stimulus-induced embedding should differ between cell types and layers. It should be, the decoding should be much better for excitatory cells compared to inhibitory cells. and it should improve from input layer to output layers in the microcircuit. Finally, the decoding should be much better for the entire cell population compared to each cell type separately. Now we're looking into the role of connectivity motif differences and also the different cell diversity that we have into and the role they're playing into shaping these embeddings. I want to thank my collaborator, Michael Rayman, and James Isbister, and also give special thanks to Mackenzie Mattis for some insightful feedback on how to use Zebra. Thank you.